let's welcome Cindy.
Hi guys, my name is Cindy, and today for Journal Club, I'll be doing the paper, The Use of External Controls in FDA Regulatory Decision Making. So just I'll give a quick introduction talking about the role of external control in FDA regulations, as well as why we need these types of controls. And then I'll go into the details about the different types of external controls that there are, as well as the current FDA guidances that exist for these types of controls. Um, so to start off, this slide introduces the critical concept of external controls within the framework of FDA regulations. So the FDA mandates substantial evidence from robust trials, and because of this substantial evidence for efficacy and safety, the FDA typically tries to prioritize well-controlled trials using internal concurrent controls. And in clinical research, an internal control is a trial design where patients are randomized into one or two or more arms in a study, and one of these arms could be a placebo arm. This type of design helps ensure that everyone in the trial is drawn from the same group of potential participants. However, there are scenarios, especially within the rare disease populations, where ethical and feasibility challenges arise within the internal concurrent control model. And this is where external controls emerge as a viable alternative, presenting opportunities not only for innovative regulatory strategies, but also in ensuring that patient safety and drug efficacy are met. So the necessity for external controls is underscored by the limitations imposed by internal controls, particularly in clinical trials involving rare diseases, like I mentioned in the previous slide, where participant availability is limited. And these limitations often stem from ethical considerations and practical realities that may hinder the design of more traditional study frameworks. So for example, in studies for diseases with high mortality or progressive morbidity, the FDA may consider external controls to be more acceptable, as it mitigates the ethical dilemma, in cases where randomization or concurrent control is not viable. Thus, you're ensuring the patient welfare is prioritized. However, in light of these challenges, regulatory approval can be a complex labyrinth. So there are significant regulatory hurdles to external control use. Therefore, it's really important to have a clear framework to show the robustness and reliability of the data used in the external controls. And this helps with understanding when and how external controls can be effectively used to facilitate a smoother pathway for drug approval, while also, again, maintaining rigorous safety and efficacy standards. Oh, next, I'll be going into more detail about the types of different external controls that there are. So according to the ICH E10, an externally controlled trial is one in which the control group consists of patients who are not a part of the randomized study as the group receiving the investigational agents. So in other words, there's no concurrently randomized control groups. And these are typically broken into different types that are characterized by time in which the subject's data was collected. So the first type of external control groups that we have are concurrent external control groups, which are based on data collected at the same time as the treatment arm, but in another setting. And an example of this is a concurrent prospective natural history data. A natural history study is essentially just an observational study intended to track the course of a disease over time with respect to variables such as demographics, genetics, environmental, and just a bunch of different types of variables. And they correlate with the disease development and its outcomes. And a natural history study may include patients that are either receiving the current standard of care or emergent care, and it can be retrospectively or prospectively done. And these types of studies help us informs us of the treatment expectations and progression over time, giving us insight into the overall treatment impact. So with concurrent external control groups, they allow us to have a real-time comparison while addressing these ethical constraints that the internal controls give us. Another type of external control groups that exist are called non-concurrent external groups, and they're also known as historical controls. And they are based on data collected at a time that is different from the treatment arm. And some of these examples include retrospectively collected natural history data, published data, as well as previous data from previous clinical studies or data from baseline control studies. And retrospective studies allow us to get insight into treatment effects without necessarily having to do new data collection. So it's a lot easier and time effective in a way. And then these are some of the current guidances that exist for external controls. And I just highlighted three of the ones that I wanted to focus on, or rather the paper really just focused on. So for the ICH-E10, the guideline titled Choice of Control Group and Related Issues in Clinical Trials, this guideline provides a comprehensive discussion on external controls, specifically stating that the choice of the control group should be considered in the context of number one, the available standard therapies, number two, the adequacy of the evidence to support the chosen design, and number three, ethical considerations. So while the guideline emphasizes that in most situations, an internal concurrent control is necessary to minimize bias and to obtain a robust statistical analysis, it also acknowledges that this might not be always feasible. Additionally, the FDA guidance titled Rare Diseases, Common Issues in Drug Development emphasizes that the product development shouldn't be delayed because of the lack of prospective natural history data. So the FDA highlights that the use of, prospectively collected natural history data is the preferred approach when you're using external controls. The guidance specifically states that the initiation of a prospectively natural history study should not delay the interventional testing that's otherwise ready to start for a serious disease with unmet medical needs. So in other words, a non-concurrent external control group may be acceptable to ensure that product developments can proceed to make sure that patients have access to treatments. This concept of flexibility is further highlighted in the recently published FDA guidance titled Demonstrating Substantial Evidence of Effectiveness for Human Drug and Biological Products. This specific guidance states that the FDA may rely on study designs that produce less certainty, such as those with external controls in some circumstances, such as life-threatening and severely debilitating diseases with an unmet medical need, for certain rare diseases, or even for common diseases where the availability of existing therapies are, they make the design choices either infeasible or unethical. And here's just a nice little table that the paper had showing when the use of an external control is most persuasive in accordance with the FDA and the ICH. And just collectively, all the guidances just reaffirm that the use of external controls is acceptable under certain circumstances, such as the ones listed here. And they reinforce the need for flexibility not only in the guidance, but also in the application during product development and the FDA decision-making process. Next, I'll be diving into how the paper tried to create a clear external control framework. I'll go into the methodologies as well as the findings and the real-world examples that they provided. So the authors reviewed FDA regulatory approvals between the years 2000 and 2019 for drug and biologic products, where the pivotal studies employed external controls, and they specifically focused on non-oncology product approvals because the use of external controls appears to be pretty well accepted in the fields of oncology. And the authors looked at the rare disease status of the approvals, the seriousness of the disease, and the degree of unmet medical need, as well as the objectivity of the primary endpoints. And they also categorized the source of the external data control just to give us a clearer picture of which type of external control is the precedent. Um, so the first thing that they found was, well, they first found that there were 45 products in which the pivotal trials were supported by external controls. And with those 45 products, nearly half of the cases were from non-malignant hematological products, with gastroenterology and inborn errors of metabolism products being the second largest category. And then it's followed by its metabolism, endocrinology, and then neurology products. And then lastly, it was reproduction, bone diseases, and urology products. And this pie chart essentially just illustrates that the experience with the use of external controls is pretty vast and varied across the divisions in the FDA. And then the second thing that they found was that a majority of the products, around 80%, that relied on external controls were for a rare disease where regulatory flexibility was given or applied due to the size of the population, or that there was a unmet medical need, or in other words, there were either no or inadequate available therapies. And the authors said that this is pretty consistent with other reports of FDA flexibility for the approval of orphan drugs. So it seems that there is like a trend where a lot of the approvals for external controls were based on whether it was a rare disease or an orphan product or not. And then not finally, but once they sorted out and characterized the external data source of the 45 products, they were able to see that retrospective natural history data were the most common source of external controls, with baseline coming in as second and published data and clinical studies being tied for third. So while prospectively gathered natural history data sources are the preferred source based on FDA guidance, none of the external controls included in the regulatory approvals that were assessed in this review were prospective. And this is really important given that the regulatory precedent is contrary to the FDA guidance, which, identifies the prospective natural history as the gold standard, while they discourage the use of retrospective natural history. So while this prospective natural history study it may be ideal. This approach is sometimes often is impractical and hard to implement, and that will lead to significant delays in the availability of life-saving therapies and could ultimately just stifle the development of products for rare diseases, which the FDA advises against in the second guidance that I mentioned earlier. Last but not least, they found that the majority of cases used objective measures as the primary endpoint. In this slide, it showed that it was 87% of the products that they studied used an objective endpoint. And some examples of objective measures that they mentioned in the article were urinary free cortisol concentration, as well as sodium phenylacetate and sodium benzoate combination. There were a few cases where the endpoint was subjective, but the difference between the treatment effect of the external control and the experimental arm were so large that the authors deemed that it was unlikely due to chance alone. Next, I just want to provide some real-world examples of all the different types of external sources that were used in the FDA regulatory decision-making. So a recent example of a retrospective natural history data as an external control source is the approval of Zolgensma, or onasemnogene abiparvovec. It is a gene replacement therapy for infantile onset spinal muscular atrophy, SMA, due to a biallelic mutation, and SMA is a rare disease with high unmet needs. So untreated patients will either die or require permanent ventilation by 24 months of age. And because this is so rare, data from only 23 patients were successfully used as an external control. And for this product specifically, the natural history of SMA was predictable, and the efficacy of Zolgensma was objectively measured to show that there was a large treatment effect. There was a 90% survival, without ventilation while on Zolgensma versus 25% based on the natural history source. And due to these factors, the FDA approved this drug as a gene replacement therapy for SMA. And another example of retrospective natural history data as a source of external control is defibrotide sodium, aka Defitelio. It was approved for the treatment of adult and pediatric patients with hepatic veno-occlusive disease, VOD, after hematopoietic stem cell transplantation. VOD is, again, a rare disease with about an 80% mortality rate, and there were no other available treatment options before Defitelio. And the primary endpoint in the Defitelio pivotal study is survival at day 100. And it was compared to the historical, sorry, it was compared to a historical control data selected by an independent retrospective review of the patient records. And they also used supportive data that came from a dose-finding study, a compassionate use study, and a registry study. And although there were some issues pertaining to the selection of the historical group, the FDA ultimately approved Defitelio based on the totality and the consistency of the data, in particular, the consistency of the survival results in the pivotal study and the supportive studies. And an example of a baseline control data being used in regulatory decision-making is in the case of Deferiprone, aka Feriprox. It is an oral therapy for transfusional iron overload due to thalassemia syndrome, which is, again, another rare disease. And the only available therapy at the time of Feriprox's new drug application review was deferoxamine. However, deferoxamine was not tolerated by all the patients. Therefore, there was an unmet medical need. So the study selected patients that were previously exposed to chelation therapy but did not respond well to it. So they were known as the refractory patients. And the selected patients were compared pre and post Feriprox treatment, and the results showed that the treatment with Feriprox significantly decreased serum ferritin in about 50% of the patients. The statistical reviewer did note that the study had a lot of serious limitations, including a lack of randomization, a lack of control group, a high rate of missing data, and they also ignored the variation between the studies by simply doing pooling. And all of these factors can contribute to bias that skew the primary outcome. Nonetheless, the FDA considered the use of a prospectively planned statistical analysis plan and the selection of the patients by an independent committee. They considered that these two factors allowed for an adequate selection of the patients for the trial, which helped reduce the possibility of bias and thus allowed for an adequate assessment of the drug effect. And so they approved the drug for marketing. And then moving on, an example of a previously conducted clinical study being used as an external control is in the case of Lamictal XR, aka lamotrigine extended release tablets used for epilepsy. And specifically for this, they wanted to add a new indication, which was monotherapy in patients with partial seizures. And for this specific study, the sponsor considered the use of a placebo or a pseudo placebo control to be unethical because there were significant control data that was already available from previously conducted studies. So they did a study design where patients who received one of two dose levels were compared to a historical control group based on retrospective analysis of control arms from 8 previous studies for other anti-epileptic drugs. So in other words, the historical control data were specifically derived from the control arm from prior studies with similar designs and methods. The FDA advisory committee that oversaw this review agreed that this historical group was acceptable under this circumstance, and this example essentially became a precedent for other anti-epileptic drugs. And then last but not least, an example of using published data as an external control is Novel 8, which is a recombinant anti-hemophilic factor. In this case, the historical control data from 9 publications were used as external controls to support its approval for the prophylactic treatments of hemophilia A, again, another rare disease. The authors compare the annualized bleeding rate, ABR, in patients treated prophylactically with Novel 8 to the ABR observed in the historical controls treated with on-demand regimens. On-demand regimens. And the historical ABR was calculated using the data weighted by the number of patients in each of the 9 published studies. And based on this comparison, they saw that there was a 68% reduction in bleeding rate when using Novel 8. And this was, again, considered acceptable for approval of a Novogene by the FDA for routine prophylaxis treatments. Okay, now I will be talking about some of the challenges, specifically some factors that could introduce bias when we're using external controls, as well as some statistical and methodological considerations that could help reduce the bias. Okay, so some factors that contribute to bias when using external controls include difference in prognostic variables. I will go into more detail about this point in the next slide. Another factor is baseline imbalances, which may also introduce confounding variables that skew the results. And another factor is the improper handling of missing data in the analysis. And last but not least, again, there is a lot of pros and cons when it comes to using whether you should do longitudinal versus cross-sectional statistical analysis. So the FDA recommends the use of longitudinal rather than cross-sectional data sources because they yield more comprehensive information about the disease onset and the progression over time. But in a rare disease setting, it's usually more likely that only a cross-sectional data source is available. And even when longitudinal data are available, these two different approaches both have its potential advantages and disadvantages, and thus you really should just be using it based on what the objective of your statistical analysis is. So for example, longitudinal data sources, you can use a lot of the available data, but it's hard to match up the clinical time points when you're comparing your external control arm versus the experimental arm. And that can result in a lot of missing data, especially if the source is a natural history source because of its observational nature. And then when you, on the flip side, with cross-sectional data, you avoid the complex longitudinal methods for handling missing data, but then you get the con, which is you have you can only use a lot less of the available data. Yes. Okay, so next, I'll talk about the statistical and methodological considerations to minimize the bias. So a major challenge in external control groups is the difference in prognostic variables that, like I mentioned earlier. And these variables can include things like demographics, diagnostic criteria, disease stage, baseline status, and concomitant therapies between the treated and external controls. And that can lead to biases, especially because we don't have randomization in these types of external control studies. Therefore, the proper selection of an external control group is really important. So there was this medical statistician named Stuart Pocock, who proposed 6 criteria that are sometimes cited by the FDA for an external control group to be considered acceptable. So from 1 to 6, it's just historical groups have had to receive a precisely defined standard treatments. Historical and treatment groups have to have the same requirements for patient eligibility, so their inclusion and exclusion criteria have to be the same for both groups. The historical and treatment groups have to have the same methods of treatment evaluation. So basically the design and methods that we use in both groups have to be similar, if not the same. There also has to be a comparable distribution of important patient characteristics, and this is talking about the difference in prognostic variables. It has to be quite similar or at least comparably distributed. And preferably, the study should be conducted within the same clinical organizations or by the same investigators. And lastly, there are no additional indications or differences that could affect results. So, and although the Pocock criteria is not applicable for all situations, it should be applied to the best extent possible when selecting a historical control group, and also to the following statistical analyses that will follow. This criteria was actually used by the FDA in the justification for the approval of Lamictal XR that I mentioned in the earlier slides because of the similar patient eligibility criteria, as well as the similar design and method of the studies. And it was, again, done by the same organization. Next, okay. Other than the Pocock criteria, other considerations include using statistical methods such as matching, stratification, covariate adjustments, and propensity scores to adjust for the baseline imbalances between the treated and external control groups. And of course, there's also an element of transparency that is really important, especially about critical aspects of the analysis, such as the exclusion of subjects and the handling of missing data. So the paper states that in order to address, missing data in non-randomized trials, you have to include a design and conduct trials that can minimize the amount of missing data, and you also have to use missing data adjustments based on scientifically plausible assumptions. And lastly, you have to collect covariate information that is predictive of missingness and the study outcomes. And a covariate is essentially just a variable that may have an effect on the study of an outcome. So that's something you have to consider too. All in all, by taking into account these considerations, we should apply just a variety of alternative reasonable analysis approaches in order to achieve consistent results, which then lends confidence to the conclusions that we find and also allows for people to trust in the findings of the studies. And when possible, consistency among results from different measurable outcomes or even multiple sources of external control data is even better and more desirable. And then just a quick conclusion to summarize the paper and the key takeaway points. Based on the products reviewed in this study, the sponsors appear to have achieved some sort of regulatory support to use external controls to push for product approval. And some factors that contribute to this approval is, despite the limitations of the external control, include the nature and the rarity of the condition, as well as the unmet medical needs, and really just the totality of the evidence. And that can include positive secondary or pharmacodynamic endpoints, or even positive data from supportive studies like we saw in the Defitelio drug. And another important point is that when you're comparing the results of this study to current FDA guidances, you realize that the statements don't really match up. So, for example, though the FDA recommends and prefers prospective natural history data as the external control, the majority of the approved products studied in this paper actually used retrospective natural history data. So it's really important for the FDA to update their guidances to emphasize that the acceptability of other external data sources, especially retrospective natural history, and also to address the statistical and methodological challenges, as well as provide recommendations for managing bias and really just to facilitate information sharing. And that is my reference, and thank you. Do you guys have any questions?